Thank you so much, everyone, for being here. It's a pleasure to be able to announce the first speaker of our annual Gogec conference. Um, Dr. Sara Molinari is currently an associate professor at University of Maryland, but she's done a lot of really interesting research in uh, engineered living materials. And so I'm personally really excited to hear her talk. She has a background originally from Rice University, where she did a PhD on cell differentiation, but has since been able to work with agencies such as DARPA to engineer these living materials. So I'm very excited to hear this talk. Everyone give Sarah Molinari a hand.
Thank you for the introduction. I just want to start by thanking the organizing committee. I'm very excited to be the inaugural speaker of this third Gogec conference. I think it's a wonderful initiative, and I pretty much agree that there's the need to spread synthetic biology and get the input from a broader community than the one that we commonly hear from, because this field is still new and there's a lot of potential. And I think the more minds we can attract to this field, the more we all gonna benefit and boost the progress of synthetic biology. So thank you for organizing things. That was a wonderful idea. Let me share my slide. Let's see if I learn. So you should be able to see that moving, right?
Yeah, we see that.
Fantastic. So I want to thank Georgie for giving me a higher rank than the one that I currently have. I'm not an associate professor, I'm an assistant professor at the University of Maryland, just for clarity. And I work on engineered living materials from bacteria. So it's probably very trivial to understand why materials are so important for the human progress, right? Like they are every object that we interact with in our daily life, both from recreational purposes or for work. Everything that we do goes through the physical contact with a piece of material. And so if you think about how important materials the human progress, you want to for example, think about steel during the Industrial Revolution, or even semiconductors for the digital era. So I'm doing in my life is I think about how to design the next generation of materials. And so we want to first think about what are the properties that the material should have, right? So for once, they should have a superior performance. So they should be able to do something that current material are not able to do, so that we can enable new application in different fields. Another very important properties that the material of the future should have is sustainability, right? Like we all know what's the climate crisis that is going on and how dire it is. And so we don't want to really keep on accumulate waste on the pile that we already accumulated. And another thing that I feel strongly about is that the material of the future needs to be affordable so that the broader community can benefit from progress as not just a classical elite of few countries or people. And so when you think about creating material that have all these three properties, it's a little bit overwhelming, right? And you think like, okay, where do I start from? And the good news is that this material really exists already, and they're like nature perfected them over the course of evolution, and they're living materials. So these materials come in many different forms, right? Here we see an animal and a plant, but there are many different forms of living material. For example, the desert crust is a living material, as well as biofilms are living materials. And despite the broad variety of forms and shapes that which they appear in nature, they all share a similar architecture. At their core, they're all made of cells that control the biological function of the material and also self-produce a biopolymer matrix that controls the mechanical and physical properties. And so why do we think that this material can deliver superior performance? And that's because living cells can do things that we're currently not able to encode into artificial material. For example, we can have the cell biosensing the environment, right? Can respond actively and dynamically to the change of the environment. So they can be made stimulus responsive. They can produce a broad variety of molecules in response of certain environmental stimuli. They can adhere to tissue. They also can stimulate our immune system. And here there's a very restricted pool of properties that, just to give you an idea, but all the performance that we can encode are the tasks that living cells are able to do, which are pretty much unlimited. They're sustainable because they're fully biodegradable, right? They're made of fully organic components. It means that this material they're not gonna pollute the environment. And they're affordable because they grow from cells with very little energy and nutrient input, right? So they just grow. It's like from a plant from a seed. We don't need high pressure. We don't need harsh chemicals. We don't need specialized personnel and equipment, which really makes expand the affordability, because this material can potentially be grown anywhere, can be transported anywhere. So there's not a limitation, like technology and equipment becomes not a limiting factor. So another aspect of the living material that I want to point out is that not only they can perform like very unique properties due to the living cells, but they can do all of this action at the same time. And multifunctionality is a property that is extremely difficult to engineer in artificial materials. It's so difficult. There are like active area of research in this field, and when the material, the artificial material, can actually encode 2 or 3 properties, it becomes extremely expensive and extremely difficult to manufacture. So just to give you an example, imagine you have a wound dressing, right? That not only mechanically protects the wound, but also fight the formation of pathogenic biofilms or stimulate tissue regeneration while also preventing excessive scarring. This is a very active field of research, and there's not a definite solution because we're not able right now to encode all the properties that I mentioned into a single material. But then we can even go beyond, right, and be creative about what we can do with multifunctionality. And for example, we can create food packaging that also tell us if the food is spoiled and produce toxins, or clothing that's gonna report on human physiology, is gonna tell us whether everything is fine with our body or not. So what my lab is trying to do is trying to re-engineer single cells so that they could self-assemble into whatever multifunctional structure we can think of. And I understand this video is made by a great artist who gave me permission to reproduce it. And it seems very futuristic, right? But in reality, if you think about the first people that envisioned, the personal computer or the phone, right, that had no idea that we could one day like walk around with a super powerful digital object. And they had to be creative because when they envisioned this technology, they had no idea whether that would have worked or not. But in our case, we know it's possible. We know it's possible because nature has already done it many different times. So our job is not to reinvent the wheel from scratch, but it's to understand how we can reprogram nature so that we can encode tailored properties, not just the one that serves the purpose of the plant or the cells that evolve naturally, but to encode properties that can help us encoding new property and novel application. How do we do that? Of course, we do that with synthetic biology. That's why we're here. And that in the last 24 years gave us the tools to really program the cells for new function. And so these are the tools that my lab uses to achieve this goal. So in my time at Rice, like you mentioned, I was part of the system synthetic and physical biology program at Rice, and then I stayed for my postdoc, so I spent a lot of years at Rice. And in this time, I actually covered different aspects and different challenges of engineering living material. So I went from design stable pattern formation that will help the prokaryotic population to expand their functionality because they can differentiate in multiple lineages, to other tasks that involves more the material as a bulk. So how we reach the microscale and tailor the mechanical properties of the bulk material. So today I'm gonna talk about mostly these two challenges, and I'm gonna touch just a little bit on what are the future direction of this field. So in order to reach the macro scale, right, we have to go from a little tiny cells to something that is at least like, let's say, centimeter big. So it means we have to encode the hierarchical assembly of the cells over 5 orders of magnitudes or beyond, right? And at the same time, we also have to be able to control the mechanical properties that results from this cellular aggregation. So in summary, what we need to do is genetically encode a synthetic matrix, which is exactly like how nature actually develops a living material. And this matrix has to microscopically assemble the cells, right? Hierarchical, like we mentioned before. But at the same time, it also has to control the ultimate physical and mechanical properties of the material. So when we talk about encoding a genetic matrix, like we want to, look for hints from nature, right? How does nature create living materials? And so mostly they use 3 different strategies that involves proteins, carbohydrates, or biominerals. And I would say carbohydrates are actually one of the preferred form that bacteria use to assemble into a microscopic structure, right? If you look at biofilm, they use different materials, but mostly carbohydrates. One of examples, the nanocellulose. And the reason why carbohydrates is like a very black matrix molecule is because it's very cheap by encoding only carbon atom. While protein, for example, they need nitrogen, and nitrogen is extremely expensive. So it's very difficult for a cell making a lot of protein. So you would think that, the natural answer is like go with carbohydrate, like make a genetically engineered matrix that is based on carbohydrates. But unfortunately, we cannot really engineer carbohydrate. They're very difficult, right? Like they're very difficult to produce because they encode, they are made with a series of different reactions. They're very difficult then to export from an organism to another because you have to reproduce all the set of reaction that have to happen in a sequence with similar fluxes. And typically when you export to a different organism, this regulation gets off balance. And because we don't have the tools and the understanding of all this reaction, it's difficult to intervene. So we're left with proteins. Because proteins, on the other hand, we know how to engineer them. It's pretty easy. The reaction that produces proteins is pretty much universal across different organisms. So you can take a protein and transfer it to another organism. You can easily engineer the protein to change the amino acid sequence. So that's, on an engineering perspective, that's much easier and is the only tool that we can effectively engineer. I didn't mention biominerals, but the situation there is even worse because we have dependence on the environment conditions. So we also have to reproduce the right environment condition. And for the record, there are excellent groups. They're working on biomineral right now. So I'll shout out to them, right? Like awesome work, but it's even more difficult. So this to say that I chose a protein-based matrix, not because it's the only option, but because it was the easier choice from an engineering perspective. But because now I'm talking to a SynBio community, I would like to say, it would be great if some of you will trying to work on engineer different kind of matrices. So the limitation, like, of course, the engineered minimum materials existed before, and it's a very active field. And so what people have done before, if you don't have a matrix that can reach the microscale, the ELMs tend to be microscopic, and then they had to be processed into microscopic materials. So that adds a further manufacturing step that then interferes with the ability to material to self-assemble. All that were based on natural living material, like the nanocellulose, and they're a fantastic group working in this space, but it's difficult to change the mechanical properties of the nanocellulose because the production of this carbohydrate is very complicated. And so we don't reach a good level of control to dramatically change the mechanical properties of the bulk material. Or they were assembled top down. It means mixing bacteria with externally provided matrices, right? They are typically hydrogels. And this is also a very active area of research where there are extreme successes because you can reach the microscale very easily with this approach. One downside of it, though, is that then they lack the ability to self-assemble and self-repair, which are desirable in the case where we want to, make the material very affordable or make them lasting very long when they work in a specific environment. So what I have done is that I start with a bacterium that is known for having like a surface layer. As a surface layer, also called S layer. It's a layer of protein that surrounds all the external surface of the cell. And we thought that would be an excellent platform for engineering a synthetic protein-based matrix because there's already a lot of protein on the outside of that cell. And there's so much protein that actually the surface layer of Caulobacter crescentus make up of 30% of the total protein content. So this protein forms a very regular lattice, as you can see here. It's made of 6 identical. So here you see different colors, but it's the same protein repeated 6 times. So this protein self-assemble into Xmer, and then this Xmer layout and cover the entire surface in a very regular lattice. And so this whole surface layer is made of a single monomer called the RSAA that has 3 major domains. Right? One is the surface anchor that, as you can see here in the picture, like attaches the protein to the outer surface of the cell so that the protein doesn't get released into the medium. There's a crystallization domain that is responsible for the 3-dimensional conformation of the protein, like the final conformation. And then there's a very, very unusual, very big secretion tag at the end of the protein, so on the C-terminus, that we already knew it's self-aggregated. And we knew because when people started characterizing this protein, they started chopping it up to see, where is the secretion tag, and they found out that if they took the secretion tag and isolated from the rest of the protein, not only will still secrete, but it will also form like visible cluster of protein. And so the problem, though, is that when the protein reaches the final conformation, the C-terminus is kind of hidden inside this protein, and so it's not able to interact with itself, right? So we really need a way to make this this piece of protein come out of the structure so that it's able to interact with itself. So what I've done is I took this protein and I replaced the crystallization domain with an elastin glide polypeptide. This is a polypeptide, like a peptide that is known to be unfolded. So in this way, with a single genomic substitution, we basically unfold the protein on the surface of the cell. Because we disrupted that domain that was responsible for the 3-dimensional conformation, now the protein is completely unfolded. It's still attached to the surface of the cell because we still have the anchor. And finally, the C-terminus secretion tag is actually exposed on the surface, right? So now it's able to interact with each other whenever it comes in contact with another peptide. So like, just to give you an idea, I kind of created a sticky layer on the surface of the cell so that when two cells come in contact together, they can stay attached to each other. On top of that, the last thing, like polypeptide, like the word suggests, it's elastic. It's known to be elastic. So when we designed this structure, we were hoping that we were also conferring mechanical properties to the overall material because of the elasticity of the peptides that we used. So we, after introducing this genetic modification that basically went from having globular protein on the surface to having like brush-like structure on the surface, we probed that with AFM, atomic force microscopy, and that I really have to credit our collaborator at the Lawrence Berkeley National Lab, the group of Paul Ashby that did a fantastic job performing in liquid AFM on the cells and show that the wild type here, see, this is the regular lattice that I showed you before. This is the cells that has the RSA completely removed. And this is our engineer strain where you see there's this like brush-like structure that projects out of the cell surface. So we also probe that with microscopy. And if you see here, I didn't mention that, but there's a little piece of protein called spike tag. This piece of protein is able to bind covalently with this other peptide called the spike catcher. And so we fuse the spike catcher to GFP. So in this way, when we incubate this material with a fusion protein spike catcher GFP, we expect that to bind to the surface of the cell. So here you see the cells in yellow. This bacterium looks like little bananas. And it's all covered by, each cell is surrounded by a layer of GFP. And this shows that actually we did successfully re-engineer the outer layer of Caulobacter crescentus in the way that we predicted. So now we have to see if it worked. And so what you're going to see right now is actually a picture of a flask from the bottom. And of course, it worked. Otherwise, I wouldn't be here today talking to you about it. And so as you can see here, the wild type Caulobacter crescentus grow planktonically, so it doesn't form any material. You see a turbid solution like E. coli. While here, the novo ELM strains, you see that it forms microscopic piece of material. And this is like a flask, a regular flask, 250 ml. So this is like roughly 8 centimeter of diameter across. So this piece of material didn't need any microscopy to be visualized, that this is in fact like a regular camera photograph. So we not only took picture with the camera of these materials because we were very excited that we could see them with the naked eyes, but we also took them like microscopy picture, like we dissect the material a little bit and put them under a microscope. And as you can see here, this material actually characterized by a huge amount of cells clustered together, exactly as we predicted. And we were very excited to see that because the most, like people have tried to engineer cellular aggregation before by re-engineering and the surface and displaying self-assembling peptides, but they could only reach a certain amount of aggregation, which was like in the range of hundreds of cells. Were here, like we went orders of magnitude beyond that limit. And so we were extremely excited to actually see this result. Another thing that I think it's really important to mention is that this experiment has been repeated. I mean, here they say 100 times, the web probably was 100 times at the time of submission. By now it's probably 1,000, and it never failed. So what does it mean that this experiment never failed? It means that this genetic modification that we introduce is not really causing a lot of burden for the cell. The cell doesn't hate it, doesn't try to mutate it out. And probably because this we didn't change the protein in a way that would have clogged the machinery, the secretion machinery, right? Like it's probably easy to export for the cell. This protein is also accessory for the cell, so it can be removed. So we are not touching any vital function. And so for this reason, it's actually very well tolerated by the cell. So now we wanted to look a little bit deeper into how this material is actually made of, like really, what's the internal structure of the material. And we've shown that on the market, like on a lower magnification, you see that this matrix, it's in blue, it spread out through the entire materials, right? You see the cell in yellow, the matrix in blue, and they largely overlap. And this is expected because once again, we know that we surrounded every single cell with a layer of matrix. And so it was obvious to observe that this matrix is pretty much everywhere. What we didn't expect is that at intermediate level of magnification, we actually saw something that looks like a cluster of protein, right? It looks like this protein are bonded together here in a region that excludes the presence of the cells. So this was unexpected because in order to reach that level of protein clustering, it means that some of the protein is actually secreted into the media, right? Like it's released from the surface, and we did not plan for it. So we're trying to, with AFM, to understand why that was happening and then how we could explain that. And we've seen that when the cells start growing, it shows like a very smooth surface, but by the time the material is formed, the cell have this like much thicker outer layer that looks like a little bit bumpy. And so we think that we may have deregulated the protein regulation on the surface. So we probably make more protein. This protein start layering up on the surface of the cell, and inevitably then the outer layer may not feel the attachment to the surface as strongly as the lower layer, and so that may be released on the media. This is speculation. This is what we think is happening. But from the picture, it seems it's clear that there's some level of protein secretion that is going on here. But the good news is that there's way more protein than we thought. So this material seems to have a lot more matrix than we originally planned for. And so we thought, okay, so it means that if we change this matrix, we should see actually a big difference in the material that forms. And so we create two additional versions of this protein, one that lacks completely the ELP, right? So you only have the surface anchor and the secretion tag, and one that does have the ELP and the secretion tag, but doesn't have the anchor. So if you looked at from an engineering perspective, it means that these two proteins are kind of similar in the sense that they're still attached to the surface of the cell. While this protein, because it's lacking the RSA anchor, is completely secreted into the media. So if you look at the material that they form, and I don't know how obvious it is from the picture shown here, but it looks visually very different. But this first two versions on the microscope actually look quite similar. You could still see this microscopic aggregation, like here, that was characteristic of the original material. But in the third example, you don't see that. See, you can see the protein matrix on the bottom that you cannot see here, and you see very few cells like loosely attached to it. So this is one thing then we learn is that if we want a cell-rich material, we really need to have the surface displayed on the cell surface so that the cells can attach themselves to this amount of secreted protein. So if you only release the material on the medium, it's going to be very difficult to trap the cell inside. And because we're trying to make a material that's going to have biological functions, of course, we want as many cells as possible. We also, took this material and said, okay, they look very different. Maybe they have different mechanical properties. And we took this material, put under a rheometer. For people who don't know what's a rheometer, I have a brief illustration here. It's made of two plates. One is like conical and one is flat. And these upper plates rotate and determine the responsiveness of the material to the force that has been inflicted. And so we calculated the storage modulus in this way, which measure the stiffness of the material. And the loss modulus does describe the viscous behavior of the material, which means like how easy it is like to break the bonds of this material and to inflict like a permanent deformation, right? So one thing that we notice is that the storage modulus is always higher than the loss modulus. And this means that they are solids. And the kind of range of values that we get there are in the range of hydrogels. So we made a material that has more or less the mechanical property of an hydrogel. Very interesting, we found that actually this material here have a 25-fold difference within each other. So being the material that doesn't have the anchor is the softest. You see like that both the surge and loss model are much lower than the other two. And the material that doesn't have the ELP is the stiffest. So now we don't know exactly why this is happening. And that's why, you know, we're trying actively now in my lab to include more material scientists into this field. What we think is that the stiffest ELM, because the ELP traps a lot of water, by lacking this ELP also doesn't retain as much water as the original material. And so this contributes to stiffness, while the softest material, because it doesn't have the anchor, it lacks some intercellular bonds that forms between the cells and the matrix. And so this contributes to him being the softest. But we will welcome other idea from material sciences that can give like a much more detailed explanation of why we observe such a dramatic change in mechanical properties. So one very, very talented student in my former lab, Bobby, actually took charge of this part of the project, and he stayed up all night, one night, to see how this material formed little by little inside the flask. Usually we observe the material in 24 hours, so it was very easy to inoculate the flask in the morning, and then the next morning the material was there like a magic. But one time he stayed up all night and he actually observed that at the beginning, it seems that the cells grow planktonically, like the wild type, but like a sort of pellicle at the water-air interface start forming. And this pellicle becomes thicker and thicker as the time goes by until it becomes too thick and it's gonna collapse on the bottom of the flask. So the explanation that we've given of this phenomenon is because, as I told you before, we think that there's a part of this protein that actually is secreted into the media. And because this protein are intrinsically hydrophobic, they collect at the water-air interface. And so this allows cells to attach to this matrix, and they cell attach more and more as the time goes by, and until this pellicle becomes thick enough that it can collapse on the bottom of the flask. So the student was really fascinated with this phenomenon and thought, okay, so it seems that the water-air interface, so this surface here, really plays a major role into making the material assemble. So what if we change that? Like, what if we change the parameter that contributes to the overall surface of the water-air interface? What happened? And so we identified 3 parameters that may have a role in shaping the characteristic of the water-air interface, and it's the culture volume, the flask diameter, of course, right? How much liquid you put in the flask will change the surface of the water-air interface, and the shaking speed. And we actually seen both in under 25 and 200 ml flasks that you can actually change not just the amount of material produced, but the form in which these materials appear, right? You can form tiny little pieces or one big piece or specks of material. And like he really described this phenomenon by converting these three parameters into a unique parameter, what we call modified volumetric powder. And it shows that this, if you change them, you can predict the behavior of the material into bigger flasks, like in 500 ml flask. And he identified like an optimal, there is an optimal level, which is like this value of modified volumetric power, where the biggest material form. And if you go too low or too high, like on the other side of this optimal range, then you're gonna have little pieces of material form. So just for practical thinking, imagine we're just changing the shaking speed, right? Like, so if we shake too high, the pellicle, it becomes fragmented, right? Because there's too much force that spreads it around. And so this, then you have tiny little micro pellicle that become tiny little pieces of material. If you don't shake enough, you don't have enough centrifuge force that force these like cells all at the center of the water-air interface. And so the pellicle doesn't become strong enough to actually hold together. And there's an optimal value where you shake fast enough to have cells and protein collecting at the center of the water-air interface, but not too much that you're going to destroy that pellicle. And that's what makes it form the largest pieces of material. This is actually an extremely nice analysis because you can use for scale up like we show. So if you want an application, you want to make as much material as possible, but you can also use it to scale down. And this is like for rapid screening, for example, right? Like if you want to prototype this material and want to make them grow in multi-well plates, having a mathematical model that allows you to scale down is actually extremely convenient. So now that we already characterized the material, we thought, okay, we can just play with that a little bit and see what happened. So we took the material out of the flask and forgot it on my bench for like 1 to 3 weeks. And we showed that despite the fact that it completely desiccated, this matrix seems to have a sort of protection mechanism that allow the material to regrow. So cells are not dead, but if you re-inoculate after 1, 2, or 3 weeks, you still see regrowth of materials. This has a very important implication in the fact that we can transport this material. Like imagine that one day we're going to have therapeutics that are based on living materials. That means that we can transport at room temperature the material anywhere and then regrow that by simply re-inoculating that in fresh media. Another thing that we did is like we showed that the material can be processed into a cohesive extrudable paste. We didn't 3D print, but the fact that we show that we can extrude it from a needle was laying the foundation for future application in 3D printing. We can also use that as cementing agent for inorganic powders. In this case, specifically, we mix it with glass powder. And I promise you that probably the cells are not alive in there anymore, but this piece of material form a tiny rock that has been sitting on my bench ever since. So the last time I checked, it was probably 3 months. It means that this material can provide an easy cementing agent that can be used on the field and can be grown out of just cells. This is a very nice application. So we know the cells of Caulobacter crescentus can actually bind cadmium. Has been shown that before through interaction between the surface of the cell and the ion. And so this is not a property that we engineer. This is a property that the cell already has. So we ask ourselves if we can actually reproduce and still use that protein in the material. And the answer is yes. As you can see here, we can incubate the material with a cadmium contaminated solution. And after the incubation, the amount of cadmium is much lower than if you don't have the incubation with the material. So this is not a new properties, but imagine you have to filter a contaminated solution. Would you rather do that with a piece of material that you can grab, steer into the water and take it out, or with loose cells, right? So this is a nice example on how materials can leverage the property of the cells and transfer that into a field-ready application, just by virtue of being macroscopic. We can also show that because of the presence of SPITAC, we can externally bind enzymes. In this case, we use glucose dehydrogenase to the material, and so that the material can act as a catalysis. And because of the extremely specific interaction between the spike and the spike catcher, the protein can be provided not just like in the pure form, but also from cell extract, so in pure form, because the material will be able to grab the enzyme out of all the mixture and use it and concentrate it. So in summary, what I've done is I created the novel engineered living materials that grow from genetically engineered cells. In particular, we use Caulobacter crescentus for its property of the outer layer. I show that this synthetic protein form actual extracellular matrix that ultimately then control also the bulk property of the material. And in particular, we measure the mechanical property of the material that we would be able to tune over 25-fold range. So I want to give you a glimpse over the future direction of this field. And so now we have a platform, right? A new class of living biomaterials. They're made of cells and a matrix. So cells can be engineered to perform different functions, like we've mentioned that before. And synthetic biology in the last 24 years really gave us a tool to encode a bunch of different properties that can be used for field application. And in particular here, it's environmental sensing, but also production of molecules. And so, and all the examples that we already observed published in the field. Another aspect that we can engineer is the matrix. So we can change that property. Like we already shown that by changing a protein, we can reach different level of mechanical property, but maybe we can even go further and change something else than elasticity, viscosity. We can also engineer surface adhesion, environment responsiveness, like physical environment responsiveness, or even electrical conductivity. Another thing that we can do to actually expand the functionality of this living material is moving into a different bacterium. Like we've seen that Caulobacter is able to bind cadmium. So that's one thing that we don't have to re-engineer, right? So we can use other properties of different bacteria so that we don't have to engineer every property that we wish the material has. One of the things that synthetic biology is exploring right now more and more are non-model organisms, because we know that we can have more with less effort. Like instead of re-engineering everything in E. coli, we can just find the bacterium that already has some property that we need, that already does half of what we wish to accomplish. So that's also a very promising area where this system can be re-engineered into different model organisms. And so I think I want to leave you with the idea that engineered living material can be extremely powerful because they increase the local concentration of cells, right? Like, so it's a system that can cluster the cells together, make them stick to each other. So having it's very cell-rich material can increase the persistency of engineered cells in a given environment, because if we want a bacteria stick to a certain environment, let's say to the roots of a plant, it's much easier to have a material like a paste that you can spread and you can attach to the roots instead of a bunch of loose cells that will eventually fall out of the environment that you wish they stick around. And also they self-regenerate and self-repair, so they can be programmed to have a much higher longevity than other materials. And so with that, I would like to take a few minutes to really mention the people that contribute to the success of the study, my previous advisor, Caroline, but also here I highlighted all the people that directly contributed to this study. Also, our absolutely fantastic collaborators at Berkeley. I didn't mention, I mentioned Paul Ashby, who was responsible for the AFM, but not Catherine Ryan. That is the guru of Caulobacter crescentus, and she taught us everything we know about this bacterium, how to grow it, how to genetically modify it, of course, the funding agency. And thank you for your attention. I'll invite you to follow me on social media if you'd like to connect. I would be thrilled to stay in contact with this community. This is also on my lab website if you would like to have updates on our research.
Thank you so much, Sarah. Really, really interesting talk. I want to hear from the audience if there are any questions. If so, you can use the little raise hand button. You should be able to. Um, but meanwhile, I just want to ask a question for myself, if it's okay.
Yeah.
So, I I'm really intrigued with functional living materials, engineered living materials, and I think one of the first things that come to mind when you say living materials is the adaptability, right? The idea that you can have cells express one characteristic of your material at one time, but then dynamically change
Mm-hmm.
according to the environment. Is that something you've looked into a lot? Creating sort of gene circuits for regulating. I know it's something you talked about as one of the benefits of using living materials, but how much have you looked into dynamically changing the properties of the material as you go on?
I didn't. This is a great direction for the field, right? So what my lab is doing right now is trying to understand the rules for bringing cells together. Like what happens if I change the magic? So we're more focused on actually engineering the material themselves more than functionalize them for a specific application. But and these are all direction that would be eventually interested. The idea of the material responding with environment, it means that we have to engineer this out. We have to introduce the circuit that is responsive with the outside. And those circuits already exist, right? So once we have a material that has the properties that we wish for, we can always then further engineer the cells to incorporate that circuit. So because we already have wonderful example of circuits that enable environment responsivity, we're not actively working on that, and we're concentrating more on the material assembling side of living material.
Thank you very much. Are there any other questions? Um, yeah, Alex?
Hey, yeah. So I noticed you were talking about some of the things that you found that that did matter in terms of the characteristics of the properties, like culture volume and stuff like that. Was there anything that you thought would matter, but when you tested it, ended up not mattering?
Yes, we trying to change the temperature. That didn't matter. It seems it didn't matter at all. We trying to change the time of incubation without like what happened if we stay the material, like if we shake the material for 48 hours instead of 24, and that didn't have any impact. Like that once the material was formed and collapsed on the bottom, it seems that no cells were actually attaching to them anymore. Like there's a threshold size that we don't know how we can regulate. So yeah, there were a few things that we tried and they didn't really make a huge difference.
Interesting.
Sahel?
Okay, thank you for the nice presentations. I just want to ask about how do you have any idea to scale it up in the mass production? I mean, is that's like a labs based on the like in vitro things on the petri dish? I think I just want to know the idea how to cultivate mass production of the that's kind of extracellular material in uh, fermentation setting or other kind of bioreactor. Do you have any something suggestion? It's that, but it is interesting idea, I think.
Thank you for your question. That is extremely relevant question. The answer is no, we didn't look into scaling up. And to be honest, probably right now, the yield of the material is pretty low. So there's really a lot of potential to trying to understand how we can increase the yield. We show with a very preliminary experiment that we can go from 250 ml to 500 ml. And it seems that the scale up stays linear. So you get a little bit more material with half a liter compared to a quarter. It's still very preliminary, and I don't know how much this ascending trajectory actually will last if you go on in a bioreactor. But that's something that we would love to explore with the right collaborators, because I think this would make the difference on whether this material can then reach the applicability that we wish for. So thank you for your question.
Okay.
Any other questions? Doesn't seem so. I have an additional question, and if it's okay. So obviously we have in the past couple of years seen an explosion in the use of AI and learning algorithms for machine learning algorithms for protein design. And so your whole living materials system is based on proteins attaching together. Is this whole I've heard people refer to it as a new era of protein engineering. Is this are these tools that are now being developed with regards to protein design something that you've considered using?
Yeah,
To
yeah,
ration
absolutely.
design?
Like this, yeah, it's a great question because it's exactly where the protein engineering is going. And so because protein engineering is relevant for the property of the matrix, AI can be a fantastic resource for us to map, like how do I increase elasticity? How do I decrease viscosity, right? Like instead of going error and trial, we can like design protein from scratch, or like infer knowledge with AI of what are the modifications that we have to introduce in order to get that desired property. So with that it's an obvious next area of research, and we're very interested in that.
Great, thank you. We are still waiting for the next speaker to arrive. So if there's no more questions, I think we can have a 10-minute break. In any case, I think you should give a round of applause to Sarah once again. Thank you so much for a great talk.
Thank you for having me. It was a pleasure to get in contact with the community, and please reach out if you have any question in the future. I'm always happy to engage with this community.
Thank you so much.